Let's continue. So now I have my other hat, not an overviewer of spatial cell types in general. Now I'm in the hat of what we do in our lab. So I'll tell you about neural codes for natural navigation in bats. So kind of what I would like to understand is the neural basis of spatial cognition, spatial memory, and navigation. And we've mentioned already, talked a lot about these brain areas, the hippocampus and central cortex, and I showed you these various cell types, the place cells, grid cells, head direction cells, and border cells. And again, typically the experiments are done in this, let's say, an experiment in place cell, an open field arena. It's not even a navigation experiment, right? An animal runs around, you randomly drop food pellets, and the animal essentially is searching for food in a random fashion. Also, you might do a linear track where an animal runs from one side to the other, maybe 1 or 2 meters in length, but it's a really very different than the navigation outdoors. So basically, kind of the questions, the type of questions that we ask is how does real-life navigation differ from navigating in a 1 by 1 meter empty box, which is the typical setup for measuring these types of neurons. And I wrote here several answers. The world is not flat, it is complex and three-dimensional. The world is not empty, it contains goals. How do these factors affect the system? The world is not 1 meter in size. Navigation occurs over kilometers. So how is that represented in the brain? And also there's group navigation, there's social aspects. So how does that factor in? So you can put more additional points here, but these are all issues that we directly address experimentally in my lab. And this is what I'll be talking about today. So the animal we work with is the Egyptian fruit bat. It's a rather big bat, so it weighs about 150 to 180 grams. Not as big as the ones in Australia, but much, much bigger than the ones in North America and Europe. So it means they can carry a lot of equipment, neural logger devices, audio logger devices, positioning tags, all sorts of stuff that you need electronics to. You also need to miniaturize the electronics, but of course, it also helps to have a 10 times bigger animal, which is what I did when I moved from the US to Israel. Instead of working with the 15-gram bat, which is the big brown bat, it's just bigger than the little brown bat, but it's not big in any absolute sense. So switching to this big animal solves a lot of technical issues. They have also very big eyes. I hope you can see that. So without me showing you anything about their visual system, you'll believe me that they see really well. They have much better acuity, let's say, than rats, for example, and also very good sensitivity for low light levels. And why bats? Actually, I will leave to the end. I think it's easier first to show you what we do, and the rationale will come easier to understand later. But really, it's about, so they are really, really interesting animals, so we would like to understand what they do, but also more generally, as I'll show you, I think they give you interesting insights about the hippocampal formation function in general. So the idea would be to understand these brain areas in general using this unusual angle, using this unusual animal that allows you to do things that are really very difficult or sometimes even impossible to do in rodents or humans. All right, so just to put you into this mindset of how do bats navigate outdoors, this was already mentioned, Eric already mentioned this in his talk on the first day. This is a study we did several years ago in wild bats. Outdoors, we equip them with these small GPS tags, very similar to the ones Anna is using and other people. And what we found is that they would generally, if released at the cave, so they live in caves, and then they go forage for fruit trees, they're fruit bats, so they go forage for fruit trees, and they will fly an individual bat. In this case, these are tracks of an individual animal over 7 nights, would fly from the cave to the same individual tree night after night, as long as there's fruits on this tree. Then it'll switch, I'll show you that later. And it's quite a nice flyway. It's not maybe ultra precise. There is, one day it may go 300 meters to the right or 500 meters to the left. There is this width of the flyway can be 1 or 2 kilometers, and also there's height. They might fly at a 100 or 200 meter altitude on one day. Another day they might fly at 600 or 700 meter altitude. This is our record is 673 meters, so they can fly pretty high, so they can see the layout of the land really far. So there is this corridor, if you can imagine, that they use to fly through this virtual corridor in midair. And so this is eventually what we'd like to understand. And as mentioned, I decided not to put the details of this study, but we've done some translocations. So we translocate them to 3 different positions. They could always home, up to 100 kilometers, they could always home back. In fact, we also manipulated their hunger. So we either released them hungry and early, 100 kilometers, almost 100 kilometers away, and then they flew really straight to their preferred tree, or we fed them and released them a couple hours before sunrise, and then they flew straight to the cave. So based on the time of the night, is sunrise coming, whether they're hungry, they could choose to go either to the cave or to the tree. And this manipulation was almost 100%, except one bat that did the wrong thing, flew to the tree instead of to the cave. All other cases, really, you could predictably say, if you feed them, they go to the cave, they're hungry, they go to the tree. So they're very, very good at navigating on these scales of a few tens of kilometers up to 100 kilometers. And you see also very straight. The average straightness index of the flight of the bat, straightness index is an index if you measure what is the direct line between point A and point B divided by the actual path of the animal. But the average straightness index was 0.97, extremely straight. I mean, these guys really go extremely straight. They know where they're going over these scales of a few tens of kilometers. So these are the kinds of things we'll eventually want to understand. We're not quite there yet, but I'll show you our route in this direction. So what I'll tell you about today is 3D spatial codes in the bat brain, particularly 3D plates and the 3D head erection cell. I'll be quite brief on that because I want to tell more about newer stuff. Then I'll tell you about vectorial representation of spatial goals in the bat hippocampus, then about the neural base of kilometer scale navigation, some kind of preliminary results that we have on that, and then about social place cells and new result that we have about representation of other animals that might be relevant for social navigation, for example. So let's start with 3D representation. This is work by Michael Yartsov and Arseny Finkelstein. Both of them were PhD students in the lab. So to start off with, when we ratified the bat, had the bat crawl in a 2-dimensional box with a white cue card, very similar to rats, on purpose, so we can compare, and using the same type of wires and everything, we found basically the same kinds of neurons. We found place cells, grid cells, border cells, and head erection cells in 2 dimensions. And many of the properties were similar. There were some differences, and one, maybe the most dramatic difference, is in this state oscillation. So I already mentioned that in the rodents, as they're running around, they have this 8 Hertz oscillation. And so if you look at a couple of neurons, in this case, you look at the autocorrelation of the spike train of 2 neurons in the rat. This is from the Moser lab. You see this very clear oscillatory pattern at about 8 Hertz. You do the same thing for the bat. It's really flat. So very, very different. And the reason it's interesting, this just kind of, I'm showing you one slide of this, is it shows the power of the comparative approach, because in rodents, it was, there are many functions ascribed to theta oscillations, but one of them is in generating grid cells. There were various theories called oscillatory interference theories that were suggesting that you can take this oscillation time and integrate it via some tricks and some movement of the animal, and then you get this oscillation in space, which is the grid. But if you see grid cells without a theta oscillation, then you can rule out this theory. So there is this theory. It's hard to disentangle in rodents, although later people had other evidence against this theory. But still, you can use bats in order to tell something general, maybe about theories of how grid cells are formed, or other theories of hippocampal function. So I think it's nice that it shows you the power of the comparative approach. You can really learn something general by using a different species. Let me move back to issue of 3-dimensional space. So if you have in 2D you have place field that look like that. In 3D it can be anything. You can imagine anything you want. So to do this experiment, when Michael Yartsov was a PhD student in the lab, we used this small wireless device that was from Neuralink at the time, a device that transmits the data wirelessly. So it's without the wires, but it transmits the data. So it works on a relatively restricted distance. This was actually a pretty powerful transmitter there, so it worked up to maybe 50 meters, but still was restricted in range, and you could still record in our flight rooms. You can record really nice spikes. So here you see the 4 channels of one tetrode, one these probes that we're using for recording, and then you see really nice action potentials, really nice spikes recorded in flight in the hippocampus. You can get really nice recordings, yes?
I don't understand one thing. In the two-dimensional space,
yeah.
Where do the bats move? In a vertical surface?
So I've done, what I showed you before, it's in a horizontal surface.
Yeah.
I've also done in my postdoc, what I've done is an almost vertical surface. You can get place cells on both.
Yeah. So they move?
It's like, you know, yeah. They do it actually quite a lot. Within a cage, if you just put a bat in our regular cages, they would, you know, move around like that over short distances, because actually flying is pretty energetically expensive. So if you only need to move like half a meter, then it would have this funny locomotion as opposed to flying. It's much more energetically.
And there is no difference in the cells, in the place cells, in a horizontal and a vertical.
I don't know. The short answer is I don't know. We haven't done the comparisons. We're recording the same neurons, vertical and horizontal. So either that or that. Yes.
So did you record gray cells in the vertical surface?
We tried. So right now we're running a study where we record from the neuronal cortex in flight. I won't have time to go into that, but I can tell you the kind of what we see. We do not see a perfect lattice, a perfect hexagonal lattice, but we do see in some neurons at least a local distance metric. So it's as if you have a local lattice that keeps to be distorted, and the difference from these perfect grids that you see, you know, in a 1 or 2 meter boxes could be because maybe 3D is different, or maybe because it's a very large environment. It's a 5 by 6 meter room, so maybe that's why things get distorted over this large space. I don't know. But now about the vertical, we tried a little bit, but you know, with this study we try to have good 3D coverage. So to have a bat both sample 3D space really well and sample 2D space really well, it's really hard. So we don't have. I don't know the answer to your question, basically. Okay, so with place cells, as I said, we recorded in the hippocampus, and we had the bats fly in this 5 by 6 meter by 2.7 meter flight room, using 2 cameras to reconstruct the 3-dimensional position of the animal, and then we got cells that look like that. So a neuron, this is the spikes, this is the kind of the firing rate map, and you see that this neuron has a spherical place field. So whenever the animal flies to a particular 3-dimensional volume of space, it might be maybe 90 or 100 by 100 by 100 centimeters or so, like a sphere, then the neuron becomes active. And in fact, in 91% of the neurons, so more than 90% of the neurons, they were spherical in shape. So really, it seems that the representation of 3D space, at least in bats, is isotropic. Same resolution in the X, the Y, and the Z dimension. So this was kind of the one-slide version, one-slide summary of that study. Then we also asked, are there 3D head direction cells, so 3D compasses, if you will, in bats? So as we already discussed this on the first day, right? That really the use that you might have for a 3-dimensional representation might depend on the situation, right? So where an animal flies 20 kilometers straight, it does not go 20 kilometers up. So really, this long distance navigation is 2.5 dimensional, and then I'm not sure what would be the usage of a 3D compass. However, inside the cave, and they have these pretty complex cave systems, I'll actually show you a picture later on, then being oriented in the 3D is very important. There's certainly no positions. They come back to the same specific positions in the cave. So then a 3D representation could be very relevant. So in any case, Arseny Finkelstein, when he was a student in the lab, asked this question. So we developed this method of recording the 3D head direction of the animal. So all the 3 Euler angles, the azimuth or yaw, the pitch, and the roll, using this kind of non-coplanar array of LEDs. Nowadays, you can do it probably better with these 9-axis motion sensors, which did not exist at that time. But in any case, we were able to record this 3D head direction of the bat, both when it was crawling inverted, also flying. And what we found is that there are 3D head direction cells, so neurons tuned to a particular direction in space. So whenever the bat's head points, you know, in this direction or that direction, some 3-dimensional direction in space, then the neuron became highly activated. And an interesting point, and somebody asked already before, what happens when the animal is inverted? So you asked, I think. Yeah, so we did that. We had animals either like that or inverted, and what happened is that the horizontal preferred direction flipped 180 degrees. It was really dramatic. Yeah, I did not include this slide, but basically if I have a preferred direction, my animal is upright, my preferred head direction tuning is in that direction. When the animal goes inverted, it flips 180 degrees precisely. So now the way we interpreted this, that this is indicative of that the representation of 3D directions is not on spherical coordinates, but on toroidal coordinates. In toroidal coordinates, you have 360 degrees of azimuth, and you have 360 degrees of pitch. So in toroidal coordinates, all of this is the same azimuth. So essentially, all of this would be the same azimuth. And then under this coordinate system, there is no flip. This is all the same thing. And when L is inverted, it's still the same azimuth in these toroidal coordinates. And then everything is conserved. And to test this idea, Arseny had actually bats do this movement, so actually crawl on the inside of a thing. And then, indeed, the neurons tend to be consistent across this firing. And again, a lot of details to go into here, but I'm trying to glossing over. Yes?
So just out of curiosity, I mean, what's the sort of field of view of these
It's
bats?
about 270 degrees.
Okay, so they could still
It's pretty
see
broad.
somewhat behind them.
Yeah, yes, yeah. So the reason this could be a useful way of representing 3D space is that it solves two difficult problems. One is the problem of discontinuity. If you have a spherical coordinate system, the classic one, then if I am a head direction cell, I like this direction, then I'm starting to pitch my head. I'm at 89.9 degrees. I'm still responding because this is still the azimuth. Then I do an epsilon further, and then I'm supposed to shut down because now it's the other direction, right? So you have a discontinuity of plus 90 degrees, or similar to minus 90 degrees. So whereas in this kind of coordinate frame, it's completely continuous. This problem does not exist. And the other big problem with 3D directions is the issue of non-commutativity. So 3D angles are non-commutative. So it's really very complicated to understand how you can, meaning that if you do, 45 degree pitch, 45 degree roll, and 45 degree yaw, it's different than the other way around. So it's history dependent. It becomes really complicated to understand how you can use a system like that. Whereas what we found, at least in the bats, is that they have a trick. When we started this project, like all the theoreticians said it's not going to work. There's no way the brain can represent 3D directions. It's non-commutative. There's no way it's going to work. So in fact, it does work. And what the bats are doing is a very simple thing. Behaviorally, they almost do not roll. They do a lot of azimuthal maneuvering. They do a lot of pitch. I'll show you that in a second. But they almost do not roll behaviorally. And then the remaining two angles, the azimuth and the pitch, they present on this toroidal coordinate, which means you have basically a ring of azimuth and a ring of pitch. So these are two independent angles, and then this is commutative. So they have this dual kind of behavioral and neural trick. If you behaviorally do not roll and neurally you represent your 3D direction on a torus, then it's commutative. So kind of this is how, at least in the bats, they've solved this problem. And just to
Just the roll thing. Do you say this for Egyptian fruit bats, or do you think all bats tend not to?
So there are some bats, not Egyptian fruit bats, but there are some bats like the Glossophagus orecina, which are amazing. Which can roll.
So would you predict that?
I don't know. There are some animals that do roll, but in fact, a lot of animals avoid roll very hard. So there are some studies also on ants that as they walk on these twigs and their bodies do crazy stuff, but their head does not roll. So most animals will try to avoid roll at all costs. And you can also give it sensor interpretations because you want to stabilize your visual input, but also this is another interpretation for that. But there are some animals that do roll, specifically Glossophaga. I mean, it's a crazy bat. They're like hummingbirds. They are nectar feeders, so they can do anything you want. They can hover in one place, fly backwards, sideways, do crazy things.
But you're saying their heads.
But they would, during landing only, they will do a roll maneuver.
But their heads, I promise you, will be absolutely horizontal. That's well known in all animals.
I'm saying there is this one bat species where they do roll, but this is kind of a very unusual situation. All animals...
The body rolls.
The body rolls.
The head will be stable.
The head is stable. So I'm saying for our bats, the head is stable, but there is one bat species that actually the head does seem to roll because they do very crazy landing maneuvers. But again, this is kind of oddball in the bat world.
Because all the birds stabilize their head.
All the birds stabilize, yeah. I used to show, like to make the point that all animals stabilize their role, there's this Mercedes ad with the pigeons where, right, some of you might have seen it, where you can take a pigeon, yeah, and you can move it sidewise, the body is doing crazy thing, you know, but the head is completely stable. So really animals maintain, avoid roll at any cost, a roll of the head. Say again?
They can't do that from day one on. It's a developmental process.
Well, this also takes time to maybe to develop. Okay. So let me just, somebody asked me, I had no movies in my presentation. Somebody asked me, can you show a movie of a bat? So I decided anyway to just for fun to show you a movie of a bat landing on a grid, just to make the point why it's important for an animal to have continuity and commutativity. It will be very bad if it didn't have that. So this is bats landing on a grid. This is slowed down 10 times. But you see they have this, they do a pitch maneuver as they land, but they really don't roll. I mean, it's hard to see from here, but really look at that. You barely see any roll. This is the bat coming in. It will also land in a second. So if they had this problem, if they had a discontinuity at plus 90 degrees, something funky, you know, in the neural circuits would have happened there. It will be pretty bad for an animal like this. Similarly, at minus 90 degrees, they take off, and it will be bad if at minus 90 degrees things would be weird in the brain. So this kind of a rationale why you want a commutative and a continuous representation. Yes?
How much difference do you have between the pitch angle of the body and the head? Would it be really different, the orientation?
Say again?
The pitch difference between the head and the body. So does the head behave differently on its own?
Oh, the body can do all sorts of crazy stuff. You see it in these movies. Yeah, see, as they turn really rapidly, their head does not roll, but this is now maneuvers in azimuth. But in pitch, yeah, the bodies are pretty flexible, so they can do almost fold over, but the head will not roll. Okay, so we've seen some. You see right here, you saw how it turned in azimuth, but really did not roll. So it really they almost do not roll. And I'm happy to say that also monkeys and mice seem to represent 3D head direction. So there was a paper published from the Dickman and Angelaki labs last year showing 3D tuning to 3D directions in monkeys. And at SFN, the SFN meeting a few weeks ago, they had a poster on mice. And really, it was extremely similar in terms of the data. They had mice upside down, upright, inverted. So first they found that they have tuning to pitch and roll, including the inverted position, the stronger tuning to pitch than to roll. As I said, they don't. So that's similar to ours. And really excitingly, they showed this new data when you flip the mouse over, also the azimuthal head direction flips 180 degrees, exactly like in the bat. So I was very excited about this. They did manipulate roll though and have a different theory. They don't like the torus model. They have a different theory because the torus, of course, can explain only these two angles, the azimuth and pitch. Cannot explain other stuff, but they have other theoretical explanations to this. But the data is extremely similar, up to even with the flip of 180 degrees of the compass when you are inverted. In fact, some of the compasses, some of your cell phones, if you open a 3D compass app, would have a very similar toroidal representation. We'll continue to show the same azimuth when you flip over. So kind of even Apple thought it's a good idea. Anyway, so now we'll switch to asking about how might you navigate to goals. So this is a project by Ayelet, Ayelet Sarel, who's sitting right there. So I'll tell the short version of her project, but if you want to ask more, you should come to Ayelet. She has also a poster hanging here, so you should definitely bug her on that. So the basic question that Ayelet asked is the following. So to navigate, we had this big debate, what does it mean to navigate? But I'll tell you what I think. To navigate is in a very simple way, is to getting the ability to get from point A to point B. And all the neurons that I told you so far about have to do with point A, right? Place cells say you are here. Grid cells also encode you are here, or maybe local distances around where you are. Head direction cell says something about north, or local north, let's say the room north. But they have nothing to say about where the goal is. Now, there is this phenomenon that I showed you of these sequences of place cells playing out, this pre-play phenomena that I showed you with population recordings of neurons. But this really has been shown maybe for real in open field environment in only one study. And then the other thing is that really you need to know the environment to do that kind of computation. And it's also the speed of this pre-play is rather fixed. So if you want to now use that to represent very large spaces, then you think that you need to stand there for several minutes kind of planning your route with the sequence of place cells. It becomes really complicated and dependent on the size of your space. Whereas another possibility is a much simpler one. All you need to do is represent the direction to your goal. And this is now invariant at spatial scale because 360 degrees are the same 360 degrees regardless of space. So this would be a very nice alternative way to represent space. So to test whether this could be the case, so before that I just want to show you that bats can remember goals very nicely. So this is some more GPS tracking in collaboration with Yossi Ovel, my former postdoc. So this is one bat that was tracked for 75 nights in a row. This is our record. Not every bat you can track like that, but just luck in this case. We had some other bats with 40 nights, but what this animal did, and I'm not showing all the 75 nights, just a subset of them, so otherwise it becomes really cluttered, but we see that it went from the cave to the same tree night after night. Then at some point the fruit was depleted, it switched to this other tree. Actually, in some cases it threw 1 or 2 nights to check maybe there's still some food in other trees, so there is some transition phase where it might oscillate between the 2 trees, but then it switches to another tree. And on night number 33 in Cyan, it did something really interesting. It started flying in the direction of this tree, but then in the middle changed its mind and flipped to this other tree. So they definitely know where the trees are. In some other nights we had this bat do, go here, then shortcut to there, then maybe even shortcut to another tree that's about here. So a lot of really interesting stuff going on, but it's just the point I want to make here that they know where the goals are. Okay, so yeah.
What's the distance, physical distance?
This is in this case it's about 20, I think 5 kilometers. Yeah, sorry, I don't have a scale bar. Yes.
How do they find food? I mean, for the first time, because
Yeah.
the trees not always have.
Right. It's an excellent question, and the interesting thing, I don't have a full answer for you because so Yossi Ovel now has a project. He's tracking young, as I think I mentioned it, tracking young bats from day one that they start flying outside with this imprinted colony, and they seem. It's really difficult because you want to track the bat and its mother and these statistics of recoveries and getting good data. So you get it's still they need to collect more data. But there are some cases where the young bat went to the same tree as its mother. So maybe it's following its mother or some other adults initially. Then and we also see this exploratory phase in the beginning where they do these exploratory flights that are kind of torturous. But when we get to the adult bats in this study, we never ever, we tagged dozens of animals here, we never ever saw a search like that. The one interesting thing, not in this bat, but we had this I'm now telling you an anecdote, yes, but take it as you will, but we had a case where a bat was flying for 2 weeks, not this bat, another one where we had like 42 days, I think, it was flying to a particular tree, and then the very first day it started flying to a new tree. It did a flight that was clearly directed but a bit curved, so you really could see like, so you can almost imagine, and then it went straight back, and then all the other nights it flew very straight. You can almost imagine that it remembers this tree because they have to know these trees from previous years. I forgot to say that these bats live very long lives, 20, 30 years, so they have many years to learn the layout of the land and to learn the pattern of when our fruit trees, the seasons of the fruits, right? So apparently to fly so straight, it has to know that from last year the fruit was there. And you could almost imagine that this curved path could have been because it didn't quite remember exactly the direction. So it was a bit off and then corrected. But this is, kind of one anecdote. But generally, the adults always seem to be flying very straight and they know where they're going. And they might have learned it as young juveniles from their mothers. But this is really in the process of checking.
I'm puzzled about the fact that they go to one tree, that they empty the tree, okay, and then they shift to another tree, but that trip could be emptied by other bats, and then instead he has food because they keep going to the same tree. So
why? No, it's an excellent question. So basically, the question is why do they choose this particular tree? And I don't have a good answer because we had, ecologists, when we were showing this, were jumping at us because sometimes you might have a tree here of the same species that's much closer and they don't go there. And this tree might be empty, no bats there, whereas here there could be dozens of bats competing for. So it's not exact. You might say they might be spreading along different trees so to minimize competition. It doesn't seem to be the case because there are empty trees in the middle. And the student, Asaf Tzohar, who did the study, went with GPS. It's really nice. You can identify the individual tree. So you went to the tree and actually tasted the fruits. So, at least the human palate, it doesn't seem to be that these are very juicy, great, sweet fruits as opposed to the other ones that are not visited by bats. So the short answer is that I don't have a good answer why they choose that particular tree. Maybe it's learned from the mother, or maybe it is tastier for them. I don't know. But one thing that I can say is that for sure it's more, it's better to fly, originally at least. Now we have a lot of fruit trees everywhere. We have fruit plantations, ornamental fruits, what have you. But if you think evolutionarily, fruit trees are not that common. They're pretty sparse, especially in winter in Israel. We have fruits year-round. These are non-hibernating bats, but there are much less of them in winter. And they have to eat almost every night. If they skip 2 nights, they're dead. So basically, the strategy of, if I have a good tree, I keep going to it night after night is a very good strategy. Yeah, but why that particular tree, I have no idea. Yes.
Are they, that was with these fruit plantations, are these natural fruit trees or are they primarily eating the oranges of the...
The oranges actually don't eat.
Okay. But then some other fruits.
Some other fruits, yeah.
That the humans are putting out there.
Yeah. I don't remember here in this particular, but it's both. Sometimes it's plantations, sometimes it's human planted trees, sometimes it's wild trees. They're pretty, they're fine with humans, but one of the reasons they're so common in Israel, they're actually considered agricultural pests, they're perfectly fine with humans. Some of these caves are not even caves. They live in cellars, in deserted buildings, any cave-like structure, they're fine with that. They get along with humans, including going to these planted trees. So, but I don't remember in this particular case. Yes?
You may have already answered this.
Sorry, I don't hear you.
You may have already answered this, but are they individually making the decision of where to go, or are they finding groups?
So it's a good question. We don't know yet the answer. As I said, the young seem to follow adults, again, based on very little data that we have. As adults, seem to be mostly going alone. You see them flying out in small groups out of the cave. So initially they're in bunches, but then they might be spreading out. But again, it could be a sampling issue because really you need to put a lot of GPS and a lot of bats simultaneously to get the simultaneous recordings, and it's tricky. So having a negative result here doesn't tell you necessarily that that's the case. But so this is as much as I can say. I don't think we know for sure. Yes?
Just out of curiosity, can you rewire the route? So say that they would go from the cave to the tree down at the bottom. They will pass a completely fine fruit tree. Can you physically place them in that fruit tree and then they will...
Yeah, we had, so one of the problems is that this is all this data is offline because this is kind of stored on a logger, on a GPS logger, and then it falls off. It's kind of glued. After a few weeks, it falls off. It has a transmitter. You find it, and all your knowledge about the preferred tree is offline. And different bats like different trees. So if we could, online, download the data, we had all sorts of ideas of putting trees on carts and driving them around and cutting trees and all sorts of mischievous experiments that never got done because we need to know in real time what's going on. Yes. Maybe last question, and then I'll move on because we didn't get to the neurons.
Exactly about this is like if a bat has his favorite tree and you chop down the tree, with the bat still go in there, circling around, understand what happens? So
I don't know about our bats, but there are studies and reports about other bat species. So for example, some of these are anecdotal. There is this report from the 60s, but it's people used to write kind of ethology journals. It's written like very storytelling way, which is really cool to read. So there's one study where there was a bat, not our species, it's a bat in North America, I don't even remember which, maybe I think big brown bat, but I'm not entirely sure, that were overwintering in this barn on a farm house. And so they were banded, and the same bat they have this migration, it's a few hundred kilometers, so they go back, or maybe there was little brown bats, I don't remember. So they went back to the same barn year after year, the same individual bats. And then one year there was a fire, the barn was completely burned down, and they now you have this huge space that was completely now covered with grass, no remnant of the barn. And these bats, when they came back a year after, they kind of circled around the exact place where the barn should have been. And there are many anecdotes like that they definitely know if something is missing, was supposed to be here, they definitely know. There are other anecdotes I can tell you from papers from more than 100 years ago, all sorts of anecdotes of this sort showing that they have extremely good spatial memory over long periods of time, of months and even years, that if they learn that they can have a barn, or they found an escape hole and then you close it, they'll search for this escape hole for, last time they've seen it was many months ago. So they have fantastic spatial memory.
Regarding this, just briefly, there was also a few months ago a PNAS paper where they had a pole and they tracked the flight routes, and then they removed the pole, but the bats behaved like the pole is still there and tried.
They still did this thing, although there was nothing there. So. Anyway, okay, let's move to neurons. So they know where goals are. It's important for them, presumably. So then let's look at neurons in small scale, admittedly, but still, do we have neurons that represent the direction to the goal? So this was basically the question that Ayelet asked, and to address this, she again used the same flat room, 5 by 6 meters, and did a very simple change, that is to introduce this goal in the middle of the room. So it's a ball, it's a landing platform where food was available, and the bats did these complicated flight trajectories where they take off, fly around, and then land. And sometimes people ask me, why do they even fly? Why don't just stay there? Because the food is right here. And I'm going to give you not a very scientific answer, but kind of what I think is going on. Basically, there are two things involved here, both of which I like. They like eating and they like flying a lot. So basically, what happens is they eat and then fly around. They'll eat and then fly around. So basically, that's why they do these things. Otherwise, if they only cared about eating, they would probably not fly much. Anyway, so importantly, they're doing these complex flight trajectories, which mean they approach the goal from various directions, then they span all possible angles, and then we can ask a question, okay, so if this is the direction to the goal, and this is the heading direction of the bat, we can define this angle, the goal direction angle, and ask, are neurons tuned to this variable, to this angle relative to the goal? And let's say if the bat is flying towards the goal, then the goal direction angle is close to 0. And then, surprisingly enough, in the hippocampus, in CA1, Ayelet found neurons that are tuned to this variable. So these are two examples of neurons that are tuned to goal direction 0, meaning whenever the bat is flying towards the goal, the neuron is firing maximally. And here is a neuron that's tuned also to this goal direction variable, but to a non-zero angle, but actually kind of to where you came from, to 180 degrees. Important to make the distinction between this and head direction, because let's say, if Eric is my goal, right? So this is now, I'm going now towards Eric. This is goal direction 0 and head direction whatever. And then this is also goal direction 0, but the head direction is flipped, right? So it's a very different thing than the head direction. Something that's egocentric or goal-centric relative to the goal. And if you look at the distributions of the preferred direction, so what is the direction which a neuron likes best, then this is how the distribution looks. There are all possible angles are represented, but there is an overrepresentation. So this will be here, and this will be there, but there's clearly an overrepresentation of goal direction 0. So more neurons that like flying towards the goal. And how does that relate to the classical place cell that we discussed in the hippocampus? So it varies. So some neurons, like this one, were clearly tuned to goal direction, but did not have a very clear tuning to place. Some neurons, the other way around, so this would be a classical place cell that does not have a tuning to goal direction, but is tuned to place. And some were both, so neurons that were tuned to goal direction, but also tuned to place. So if you look across the population of neurons, we have lots of place cells and a population of goal direction cells, about half of which are also place cells. So there's kind of a mixed selectivity for these two variables. Then Ayelet did an experiment that I think is really a crucial one. Because so far what I told you is that there are neurons that respond whenever I'm going there and Eric is whatever, 45 degrees to the right. But this could be just visual, right? Any visual neuron in any visual cortex area would behave like that. So far it doesn't relate to navigation. It just could be a visual response. So then what happens if we block the goal and the animal cannot see the goal? So this is the next experiment that Ayelet did. So this is again a top view of the room. And Ayelet put this curtain. It's opaque to both vision and echolocation. And then in the first session, the goal was here behind the curtain. I mean, it could be in random position. Every day, it was in a different random position, either here or there or here. And then the animal was flying around and measured the goal direction, the tuning to this unseen goal. Of course, the animal, after flying around, then it landed here. So it then knew that, okay, today the goal is here. So potentially it could know where the goal is. And then the second session, I had to move the goal to the center of the room. And these are some example cells to what she found. So these are 3 neurons that were tuned to the hidden goal behind the curtain when the bat was flying here. Okay, they're clearly tuned to the hidden goal through the wall, through the curtain. And when computing the tuning to the same position in space, but when the goal was not there, in the second session, the neurons became almost flat. And conversely, this neuron is a neuron that likes to respond to the central goal was in the center position, but it goes completely flat to the same position in space when the goal is not there. So from here you can see 2 things. One is that there is a representation of a hidden goal kind of through the wall, which I think is absolutely crucial, because at the end, navigation is about, the interesting part, there's of course beaconing, which Eric mentioned. When you see your goal, you see the mountain, you just go through it. But this is kind of the most trivial part. The really interesting types of navigation are where you do not see your goal. It's occluded. I know that the coffee is over there, right? I don't see it, but I have an arrow in my brain saying the coffee is about in this direction. So this is the interesting kind of navigation. And so for that, you really need to find neurons that will represent cognitively unseen goals. And the other thing is that we have this dependence on whether there is or isn't a goal there. Yes?
Have you moved the hidden goal so it's still hidden but isn't hidden?
No, we haven't done that.
Do you have a prediction about what might happen if you did?
It's in the plans. We plan to change things and to manipulate, to have multiple goals. But yeah, we can talk about this. But presumably, once the animal, if you move it and the animal flies and it's in the wrong place, then the tuning might change or shift. But you can come up with all sorts of your guesses are as good as mine here. Yes.
But then what would you
sorry. No, it's okay. It was this. You were in the same vector from me, so yeah.
Okay. What would you expect to happen with 2 goals, for example? Is the animal responding in the same way to the 2 goals? Or they are just selecting one, and then you have selectivity for one goal, and then one neuron is selected for the other?
I think it's, we plan to test it kind of in the next project with Ayelet, but it could be that, yes, that they are presenting one goal, or maybe the current goal I'm navigating to, and then when I change my mind and switch to another goal, then these representations, these vectors might switch, which would be a really cool kind of readout of the animal's attention or planning in a way. So this is something we plan to do. Right now I don't know the answer.
Yeah, my question was in a similar direction as to because in some way you think that the animal, if it's foraging sites or something, want to keep track of, maybe if they are reoccurring, right? If the kind of foraging it does is you want to return to a previous site, but temporarily it is not interesting, whether you have this kind of memory, where it's like a stable.
You mean for more important locations? Yeah, it's an excellent question. Whether more important, obviously each one of us knows hundreds of places in our hometown, right? So at some point there needs to be some overlap between the neuronal populations, and maybe there's some special coding for more important places. All of these are open questions that we have no idea what the answer is. Right now we're looking at one goal at a time. Now another useful variable to have is a distance to the goal. So here I'm showing you examples of 6 neurons where what's plotted here are the rasters of individual flights, so 96 flights to the goal in this case. And this is the path distance in this case, the distance along the path of the animal. And here you see these two neurons really fired, the black dots are the spikes, of course, and here these two neurons fired really strongly, very close to the goal. Whenever it really was getting there, it fired strongly. These neurons fired a bit further away, and these two neurons are pretty crazy. They fired like 7 meters before the animal arrived. They had this clear tuning, but 7 meters before the animal arrived. So they really represent various distances. If you look at if you now turn each of these tuning curves to distance to a line here, then we had more neurons that represented short distances, so 0 to 3 meters or so, but we had all the other possible distance being represented at the population. Yes?
Isn't that absolutely crazy? Because your room is not that big.
Yes, I agree.
So like, that means that it's only possible
yes.
if the bat has planned the whole bloody circle, it's going to fly
the whole body circle.
In advance.
Yeah, it's about 2 circles or so. Yeah, okay. Because the room is 5 by 6 meters, so that means that, you know, you plan, okay, I'm going to do a couple more circles and then land. Otherwise, it's impossible. I agree. Yeah, it's pretty crazy, but it works. If you look at the, yeah.
Have you tried also to do the same analysis but with time? So the time.
Yes, we did that, and
did you observe some kind of prediction?
Well, I'm not sure about prediction, but we looked at the same kind of tuning curves with time and obviously distance and time are very strongly correlated here, but the tuning was a bit nicer to space than to time. But it was also obviously representing time to landing as well because they're correlated. It's in one of the supplementaries of the paper. So again, if you look then at the, I'll just finish this. If you look at the relation between these various tunings to place, to goal, and to goal direction, to goal distance, and to place. So this is how it looks. We have a lot of place cells, goal direction cells, and goal distance cells. And again, it's a mixed selectivity. We have neurons that are tuned only to this, only to that, but then to various mixtures of the variables, as you see here. And maybe I'll just finish because I have only, I think, 1 or 2 slides for this story, and then we can have more questions. Then, in particular, if you look, let's say, at these neurons that are both tuned to both goal direction and to goal distance, then you can plot these conjunctively. So this is an example of a neuron plotting the firing rate versus goal direction and path distance. So you see that at short distance from the goal, this neuron was nicely tuned to goal direction 0, but then looking backward along the path distance of the animal, it's still tuned to goal direction 0, but the firing rate goes down. But looking at that, you see that it's clearly representing conjunctively both the direction and the path distance to the goal. Now, most neurons were tuned more strongly to these path distance variables, so along the path, but there were some neurons that were better tuned to the Euclidean distance to the goal. So really, the straight line direction, which would be then exactly vectors, right? Okay, so to summarize, we found this story found in the hippocampal area C1 a subpopulation of neurons tuned to egocentric goal direction. The results from current experiments suggest that the goal direction tuning is memory-based or mental, not just visual or just sensory. The goal distance sets represent the distance to the goal. And then together we have this vectorial representation, which I'm very excited about because for several reasons. One, it could directly support goal-directed navigation because you have here a variable that's literally encoding the direction. So if you read that out, it kind of gives you the instructions where to go. So this is as simple a representation or as a direct representation as you want. And the other one is that, of course, it links to a lot of what we heard about in the last couple of days because this concept of home vector has been around for 70 years or whatever. Probably people will correct me that already Aristotle said that, but anyway, I know at least 70 years. And so it was shown in insects, all the way from insects to humans, that animals can do vectorial computations when navigating, but what is the neural basis of that was unknown. So these are the first neurons that might be supporting this kind of a home vector representation. And lastly, I also want to say that preliminary results indicate that there are similar vectorial egocentric representations also in rats. There were a couple of posters in SFN a few weeks ago, one from the Meta lab, another one from the Knirem lab, basically finding egocentric representations in C1 and LEC. They were surprised to find about each other, discussed it, and they already decided to submit back-to-back, whatever. So, but they were both finding similar things. Not so much in MEC, actually, but in medial neuronal cortex, but more strongly in lateral neuronal cortex and in CA1. And I think I would be fascinated to hear in the Drosophila whether you find these representations, because insects clearly can represent directions to goals. So if that's true, then this is really amazing. This means you have the similar representations from insects all the way to mammals. Okay, then before I switch to the next story, any other?
A few slides ago, you showed that you have like a lot of neurons that are responding to a very close distance to the goal. It looks like So it looks like you're increasing the resolution once you approach the goal. So is that coupled to, for example, an increase in the frequency of echolocation?
Yes. I mean, we haven't measured it in this case, but I bet it is, because we know that as they're coming to land, they increase the rate of echolocation. So it could be in parts. Yeah, so it could be sensory driven, but it could also be kind of importance, more cognitively. You know, it's hard to dissociate these things. But surely they increase the echolocation rate as they come to land. Yeah.
So you're saying that for head direction cells, there are many different populations of head direction cells in different places.
Yeah.
Do you think that they might just be these goal direction cells, or in respect with head direction to different goals in their environment?
So, I mean, it's hard to tell, because in principle, if you have a goal direction tuning relative to infinity, to something at infinity, it's like head direction tuning, right? So, and it could be, because some of these experiments are done with, you know, you have really the big room, you have a door that's effectively almost at infinity. So it's hard to dissociate. Really, to dissociate these things, you need to do an experiment similar to what Ayala did, that is to have the goal in the center of the room and approach from various head directions, and then you could dissociate these variables. Otherwise, if it's tuned to some landmark out there, then it's impossible to tell. But it could be, yes.
Do you think it's more egocentric or allocentric navigation?
I mean, it's egocentric in the sense that, by definition, it's tuned to the direction relative to the goal. So, or egocentric or goal-centric, I mean, whatever, it's just names. But it's clearly not allocentric in the sense that it represents an absolute direction in space. It's direction relative to the goal. Yes?
I would like to ask, all of these data from different cells from a single individual, or the different cells from different...
No, these are, don't remember about these 6 examples. I think they come from different individuals, but in general we had several animals in this study, so it's not just one animal.
Okay, and the other question is about whether there is a cell distribution pattern in this hippocampus.
Cell distribution meaning?
Something like this is a different path distance to goal cell, they distribute in the different...
Like you mean in the brain, yes. Are they mapped? We tried to look at that a little bit, and it didn't really find anything. So, but you know, again, we had the recordings, so we have 4 tetrodes, so, you know, not all of them have good signals, so you can usually compare like one tetrode to the other that are relatively close by. I mean, these things are better to do, let's say, with imaging, where you really see the spatial layout. So, but anyway, from what we did, we could not see any clear differences, which is different from, I don't want to have time, but I have another story at the end. There we do see some functional anatomy differences. Well, I excluded the slide here. Yes?
What is the frequency of these different cell types in these different areas? So when you put in tetrodes and stuff like that, so are these all the cell types in that area?
No, we have also cells that are neither one.
Yeah,
but
so what's the percentages approximately? The
percentage of the, so let's say the place cells, I forget now, 33%. It depends on the environment, but in this case, one third. So the pink is 33% of the total. Okay, but then, you know, overall, about half of the cells did something, one of these things. Yeah. Okay, maybe, yeah. Okay, you, and then I'll move on because I have 2 more stories to tell. Yes.
It's also more general and a bit naive, maybe. So, for example, the place cells seem to be a bit randomly distributed, and I know it's very difficult to test, but do you think that both hemispheres have a similar orientation, and then when you come to goal direction cells or head direction cells, and you think about something like the central complex in insects, do you think there would be a central integrator which does both sides?
So we only record from one side, from the right hemisphere. And in fact, a lot of the hippocampal studies are done in only one hemisphere. There are some labs to do bilateral. There aren't really, in general, I would say that in mammals, you don't find very strong lateralization, unlike in birds, for example, or in humans. I mean, humans are kind of unusual. We have these Broca areas, etc. But in general, in mammals, you don't see, you know, in rodents, for example, where people have looked at that. Some years ago, there was a study in Science or Nature from motor cortex. I don't remember which lab it was, where they found like 5% difference in some density of axons between left motor cortex and right motor cortex. And it went all the way to Science of this 5% difference because, yay, we found a laterality there. But really very little is known in general in the brain of mammals about difference between the two hemispheres. So we haven't looked, but I don't know. But
so would you expect basically to have a similar pattern for one room on the right hemisphere since the sensory?
Probably. Those people who do record on both sides, Matt Wilson, David Foster, people like that who record simultaneously, I don't think they've found any major differences. And so you see kind of all possible positions here and all possible positions there. In fact, these two hemispheres are extremely strongly connected and very strongly correlated. The two hippocampi are very strongly correlated. Okay, let me it's okay, I'll move on to tell you briefly about a little representation of large-scale spaces, and I'll tell you about the new story that we have on the social side. So this is work by Tamir Eliav, a PhD student in the lab. So, you know, the basic question is that, right? I've showed you that these bats navigate long distances. So now if you would like to take your 10 centimeter place fields as you record in the box in the laboratory and use them to tile this kind of space that's, you know, 15 or 20 kilometer long, 2 kilometer wide and half a kilometer high, this is roughly the size of it, then, and you need several cells covering one location, then you do a quick back of the envelope calculation, you come up to about 10 to the 15 neurons or so that you need. Now, entire dorsal hippocampus has about 100,000 neurons, so it's 10 orders of magnitude off. There's no way that what you find in small boxes in the laboratory will translate exactly to the outdoors. It's just impossible. So something else must be at play. And so to address this question, and just to say that it's not just that outdoors they fly these distances, but also in cave systems. This is an example of one cave system where there's a colony of bats where they have to fly hundreds of meters. You know, there is one exit here, another one there, so it's pretty complicated, the maze there, and they really have to learn and to represent this space that's underground, that's hundreds of meters in size. So, and we really don't know how that works in the brain. So to address this, we need to do three things, technically. One is that we need to be able to record in principle at an unlimited distance. So the telemetry system I told you before that was transmitting the data, that can work up to a few tens of meters, but not over long distances. So we developed this system, this neural logger system that records the data on board the animal. This is now 16 channel system, but hopefully soon Ayelet will have a 64 channel version. It's also very lightweight. This is how it looks on the animal. Now we have actually a system that's even smaller than this, almost half the size of this. And so this records the data on board. So in principle, you're unlimited in distance now. The other thing you need to solve is localization of the animal, because GPS is not good enough. Why? Because if we want to differentiate between representations that are 10 centimeter in size versus something else, we need to be able to measure the position of the animal with 10 centimeter accuracy over long distances. GPS, regular GPS, gives you about 10 meters or 8 meters, whatever, something like that, the accuracy, which is 100 times too bad for us. There's differential GPS, but that's still not quite small enough to fly on a bat, although it's getting there. So I think in a year or two that'll be available. But what we did is this is a solution from a French company that kind of, instead of, the reason GPS is not precise is that you measure differences of times of arrivals to satellites in outer space, and you have atmospheric aberrations on the way, and this is what gives the inaccuracy. Whereas if you do the same thing with a ground-based array of antennas, then the distances are much shorter and you can measure positions much more precisely, and this system gives indeed about 10 centimeter accuracy. And the third thing is we want to have a controlled but very large behavioral setup. So we can have the animal fly multiple times to the exact same location, but have a controlled experiment. So we've built this tunnel. It's 200 meters long. This is the very end of it. It's actually much longer than this. And we put landmarks, various objects. So for a while, Tamir was walking around campus and kind of collecting a junkyard of all sorts of abandoned tables and like thrown out bicycle racks and stuff. And so we've put all sorts of physical objects in there in a random fashion once every 20 meters or so on average, but kind of roughly randomly dispersed. And so this is how it looks. We have this tunnel that's 150 meter this arm and then 50 meter that arm. And we plan, hopefully this year, to build a much longer one. It's almost 1 kilometer long, and also we have plans to build this complex flight maze that looks very similar to this, but it's really a maze structure, and then we can start asking questions on more complex structures. And then I'm going to show you just a few examples. So first of all, in terms of behavior, what Tamir did is put basically balls with foods at both ends, okay? So the bat flies back and forth, and really you see if this is 200 meter long and this is the 100 minutes of recording, the bat is flying like crazy, back and forth, back and forth, back and forth. I mean, my main worry when we started this project was, and what if the bat refuses to fly? What are you going to have to do, right? It just flies and then lands and not cooperating. Will you kind of take electrical bikes and start riding after it, and so, which we do sometimes in training, but like it becomes a practical issue. What do you do? But basically, on the first time we released them, our worries were erased, because basically, I mean, you do need to get them acclimatized and get used to this, but they really love flying. They're just flying back and forth, back and forth. Some of them don't even eat that much, or certainly not every landing they would eat. They really love flying. You give them this enormous space, and they just fly back and forth, back and forth, back and forth. Sometimes 100 laps or even 120 laps. So we're talking about 20 or 25 kilometers distance in an hour, 1 hour, 1.5 hour session. But they also fly very fast. So in this case, also very stable. You see very stable speed. In this case, 7 or 8 meters per second. We had some days and some bats that fly more than 10 meters per second. This is 40 kilometers per hour. This already gets very close to what they do outdoors. So really, it gets close to the real thing. And then this is an example of a neuron that Tamir recorded. So what you see here are rasters separated by the blue direction, which is flying east, and the red direction, which is flying west. And you see each line here is an individual flight, and dots are spikes. And what you see are several things. First of all, you see that the map for the east direction and the west direction are different. So this I already showed you. This is an old news. We know that. But then there are some surprises here. So first of all, you see that there are really nice, reproducible multiple fields in this environment. It's not your single Gaussian field, but multiple fields. Moreover, some neurons had huge fields. Like this field is almost 30 meters in size. This is dorsal CA1, where you're supposed to have 10 centimeter place field, but we see some neurons with 30 meter place fields. And the most surprising, at least for me, is that we see neurons that are multi-scale. So you see this neuron here has a really large place field here, but the same neuron has a very small place field over there. And this is another example of a neuron that in this case does not fire at all in one direction, fires only in 2 locations in the other direction, but again, has a small field here and a large field there. A lot of these neurons, we have cases of neurons whose fields have the same field, 2 different fields for the same neuron could be up to 15 times in scale. So it's really a multi-scale representation. Not all neurons do that. This is an example of a neuron that has multiple fields, all of which are roughly the same size. But in general, we have diversity of firing patterns, but in generally, we have a lot of these multi-scale neurons, which is very surprising, at least for the hippocampal crowd, because there are many models of place cells that are very different from each other, but there's one thing that's in common to all of them, and that is they kind of either assume or impose a single spatial scale. So a single sigma that kind of characterizes the size of a neuron. So having the same neuron with very different resolutions, different location, that's very surprising, at least for our kind of crowd. Yes?
Is there a change after the change 90 degrees?
So we see some stuff. I mean, this is all preliminary. I mean, it's not so preliminary. We have already, almost a couple hundred neurons recorded, so it's not entirely preliminary, but we see, we're still collecting data, but we see at the turn point sometimes neurons turning off. So like interneurons have like these notches, so there's maybe a remapping or something funky happening at the turn point, but I don't really know much more than that to tell you. Yeah, okay, let's go from...
I just have to ask, so can you actually use, so you have this tunnel,
yeah,
and you can also make a miniaturized version of this tunnel, and just track the same amount of neurons and see, so if the same place cells now behave in a different way.
It's in the plans. So we don't have right now, but what we plan is when we build this longer version of the tunnel, it's almost 1 kilometer long, we'll have also a short arm that's almost only 12 meter long, and then ask this multi-scale question. So are the same neurons representing a very long environment, it's 800 meter long, and also a very short one, or it's different population of neurons? The neurons have same field size in both, or do they rescale? These are all open questions, but we plan to do that. Yes?
Is there some correlation between base field size and the speed the animals travel?
It's an excellent question. There are indications in rats that there is, that if you run faster, place fields might increase. But in fact, that's why I wanted to point, I went to labors to make the point that the velocity here is extremely fixed. I mean, there could be plus minus half a meter per second, so like 10% differences here. But the field sizes vary between here and there 15 times. So certainly differences in speed cannot explain this. But yes, there is generally, there can be a relation like that. That cannot explain what we see. So yeah, these are just examples. One thing I also wanted to mention that I forgot, but during the break, it's kind of something that's so obvious for us that I forgot to tell you, but all of this, all of what I showed you so far was done in wild-born bats, okay? All our studies, we go catch bats. I'm the main catcher myself. We go bring bats from the outdoors. So all the studies that we've done so far will be wild-born bats that have seen the real world. We are now starting to we have another project where we start to look at this tunnel in wild-born bats that have seen, obviously, kilometer scales versus laboratory-born bats that have never seen a kilometer scale, only seen a small room where they grew up. Will there be a representation? And we see interesting differences. It's kind of even more preliminary, so I don't want to go into the detail, but we're starting to look at these differences in wild-born versus laboratory-born. But everything I'm going to show you today is from wild-born animals, which is, I think, is also an interesting question, because we have no idea how are these neurons firing in wild-born rats or mice. I think it's a basic question. I would like to see real wild mice or wild rats recorded. I mean, is it the same or not? I think it's a very basic question in my mind. All right, in the last few minutes, I'll tell you about our last project. Not the last, but another project. The last project I'm going to tell you about today. And that's about social place cells in the bat hippocampus. This is a study by David Omer, a postdoc in the lab, and together with Lior Alas and Shir Maimon. And so, the basic point is that bats are highly social mammals, and they definitely know where are other individuals in the cave, where are the alpha males, they keep away from them. They have this social awareness, and also possibly they, in some cases, navigate together. At least when they're young, it seems to be evidence that they are navigating together. So it'll be interesting to see in the hippocampus, just like there are place cells representing where I am, are there neurons that are representing where you are in space? So this is what David looked at. And so we devised this very simple and a bit of an artificial task, but it allowed us to really get at this in a very controlled manner. So the task is very simple. We have two bats, a demonstrator bat and an observer bat. The demonstrator bat has a blue LED, and its task is to fly to either ball A, in this case it flew to ball A, or in some other cases it flew to ball B. So it's kind of randomly, it's supposed to randomly, it's a highly trained animal that knows to fly randomly either ball A or to ball B. Now the observer bat, which is the one implanted, you're recording from the hippocampus of this animal, has to mimic the other bat. So if, in this case, the demonstrator flew to A, then the observer also has to fly to A. If the demonstrator flew to B, the observer also has to fly to B. So this ensures that they have to pay attention to the position of the other animal in order to perform the task. And in addition, as you saw, we created this situation of an alternation. When you are flying, I'm not flying. So this will allow us to dissociate between self-movement and movement of the other. And moreover, there was spontaneously this delay. So the other bat would fly, come back, and then on average, the observer bat would wait on average 12.7 seconds before it took off. I mean, we did not impose it, there was no barrier or anything, but just spontaneously, this was the average delay, which in fact is quite a long delay, meaning not only have to have a spatial attention to the position of the outer bat, but also spatial memory. You have to remember for an average 12.7 seconds where did the other one go so you can mimic it. Okay, so we try to create this situation where you really have to pay attention to the spatial position, remember the spatial position, maybe we will find a representation of the position of the other. Okay, so I mentioned the spatial attention, and we created this behavioral space clamp. It's very important. So when you're flying, I'm not flying, because obviously, and we also had this, I'll show you this in a second, we had this accelerometer or 9-axis motion sensor on the head to rule out any micro movement, because you could always imagine as the other one is flying, maybe I'm moving my head a little bit, and this is the hippocampus. Yeah, you might be moving your head through micro place field or something like that. So it's a big caveat that you have to take care of. So we had this accelerometer to rule this out completely. Yes?
So how do you train your animals to do this?
So the trick here, there's one important trick, and that is relates to this alternation. This is something completely non-trivial. Why don't they fly together? And the reason is, the chief reason is that the demonstrator bat is an alpha male. We try to pick them such that it's an alpha male, the more dominant male. And so then basically, you know, when you're flying, you know, I give respect. I don't mess with you. The other way around, the reason it works is that these demonstrator bats were highly trained. We basically pick for that the best bats, the smartest bats that can learn the rules of the game. So although they're alpha males, so they can fly with the other, you know, they learn the rule and they know what to do. But really, having an alpha male was crucial here.
How do you motivate them?
They get food, sorry. So when they do the right thing, they get food.
They are food deprived?
You know, not even that much food deprived. In fact, we find that, you know, they don't have to be very hungry. They're a little bit, but it's not like you need to very seriously deprive them. A little bit, but, you know,
Is the upside down bat tracking the other one with primarily vision, or are they also?
Both. So we recorded, and I'm not showing that, but we actually recorded in a couple of these animals, or the couple of these pairs, we had also ultrasound recordings and recorded collocation, and they do emit echolocation calls. And also there's light available. So in this case, they could use either vision or echolocation, and I cannot say which one they use, but potentially they could use both. We did not try here to control you know, for which sense they use. Okay, so to show you an example neuron, so this is a neuron that, so on the left is, the classic self-representation. So these are my trajectories with my spikes. Yeah, I'm an observer, so this has a nice place field here, but when you plot your trajectories with my spikes, then you see that it also emits some spikes encoding the position of the other animals. Now, in some cases, you know, in many of the cases, these neurons were both classic place cells and social place cells, as we call them, representing the position of the other. This is another example. When I'm flying, then the neuron fires here. The same neuron, when I'm stationary and you're flying, it only responds when you're on the right arm. So again, representing the position of the other animal in space. Some neurons only represented the other animal and did not fire at all for self-movement. So this is kind of an extreme example of a neuron that did not emit a single spikes when I was flying. So it's not a place cell, and this is, but it did encode the position of the other animal. And this is not a stability issue, because really it's completely alternating. You are flying, and I'm flying, you are flying, and I'm flying. So it's completely intermingled, but still no spike here, and this neuron is representing the position of the other animal when the other animal is flying. And of course, we found also vice versa, classical place cells like this one that represented very nicely my own position, but did not represent the other. So again, we get this, you know, all possible combinations, and again, we have this kind of a mixed selectivity, if you will, where some neurons, a lot of neurons are place cells, and there are these, about 18% of the neurons are social place cells, and a little bit more than half of them are also place cells, and half are not. So we could rule out, and again, we did several controls. I think for lack of time, I'll start skipping the controls, but we had this accelerometer so we can make sure that, you know, when I'm flying, of course, the observer is flying in red. There's the velocity of the observer. There's large acceleration signal accelerometer because I'm flying, so obviously that you expect. But when the other bat is flying, it's really flat. And we also identified those cases when, I did move my head when you were flying and excluded them, and there's really no change in the place fields, whether I exclude them or not. These are 3 examples, and across the population, you look at the correlation of the map using all the data versus the data excluding those cases when I did move my head, and there's no change. So really, self-movement cannot explain this. It's really representing the position of the other bat. You could also say, okay, maybe it's not really representing the position of the other bat. Maybe it's trajectory planning, because I'm seeing you flying, but what actually I'm doing is I'm planning my own flight. I showed you these trajectory sequences. So to rule that out, what we did is we compared the correct versus incorrect flights, because if I'm incorrect, then presumably the bat did one thing, but I was planning something else, and we should see a difference. But in fact, we did not find a strong difference. These are 2 example neurons showing a similar firing, similar place field, similar social place field, similar representation of the other for correct flights versus incorrect flights. And when you look at the correlations across the population, there are clearly reasonably high correlations there. So it doesn't seem to be trajectory planning. And we have some other arguments against that. These 3 different arguments. I'm just going to show you one. The other question that we asked is this representation allocentric or egocentric? So what we did here is we asked, okay, we could, so as I said, from time to time the animal did move its head. It was not correlated to the movement of the other bat, but it was hanging around, and sometimes head was pointing in that direction, sometimes in the other direction. So we could just take all the head direction, behavioral head directions by the observer bat, and just split them in half. So find the median head direction and just compute the representation of the other when I'm looking right versus I'm looking left. And again, you see in these 2 examples, the maps of the other's position when I'm using the right-looking trials versus the left-looking trials are quite similar in these 2 examples, and also across the population, it's clearly shifted to the right. So kind of arguing against an egocentric representation, suggesting that there is an allocentric representation of the position of the other. And lastly, I'm kind of hitting the, the gas a bit here. Lastly, what we asked is, okay, is it really for another animal, or is anything that moves out there, any object, will elicit it? You can imagine that there is this virtual receptive field, and that anything that will cross it will activate the neuron, right? So we have to check that. And so to do this in 2 of the 4 bats that we had 4 pairs of bats. So in 2 of the 4 pairs, we had 2 additional sessions. So in addition to the session that I showed you before, when there is a demonstrator bat flying and I'm supposed to mimic it, we also had a demonstrator object. So literally an American football that's being moved on a stick, either to ball A or to ball B, and I'm supposed to mimic it, and then I get rewards. So it's the same task as with the other bat, but with a plastic object. And then a 3rd session when another object, the cube, was moved, but that bat did not get any rewards, so it did not fly. So this is kind of an important and relevant object, and this is a not so relevant object. And what we found is that this is an example of, for one neuron, that in fact is representing these 2 objects, turns out. So there is a representation in some of these neurons of the position of a moving object, but you see that these 2 objects, the informative one, which I'm supposed to mimic, and the non-informative one, the 2 maps are quite similar and different from the representation of the conspecific, of the other bat. And so the 2 objects represented similarly and different from the other bat. These are 5 more example neurons where you see in all of these cases there are similar representations. These are the bat and the 2 objects, the other bat and the 2 objects, and you see that you have similar representation for the 2 objects and different from the other bat. In these cases, this neuron actually did not represent the other bat, but did represent the 2 objects similarly. This neuron does not respond to the object, but did respond to the bat. So you see that generally the 2 right columns are kind of similar to each other. And this is captured by the population where, indeed, if you compare the maps to the 2 objects, these 2 maps and the correlations are significantly larger than we compare this to that or this to that, which are these 2 bars. Okay, so there is a clear difference between the representation of another animal and the objects. Lastly, there was also an interesting difference in the directionality. So we did find that if we compare the map when flying to the object versus back, to the landing ball or back, the map for self-flight was completely orthogonal. So this red bar is looking at the correlation of the two maps when I'm flying myself. For the self-place, classic place fields, when I'm flying there and back, the correlation is 0. Okay, so this is these old news that place cells are directional. But it was also close to the 0 when the other bat was flying or coming back, but it was actually pretty high for the object. So for the object, there was really not much of a difference between the two directions. They were similar to each other. And again, you can give it a social interpretation that if an alpha male is flying away from me or towards me, these are two quite different things. Whereas, objects are maybe not so important. So there is this interesting difference. Also, I glossed over, we had somebody asked about functional anatomy. So here we did find an interesting difference in functional anatomy, that place cells were more concentrated in the proximal part of CA1, which was shown, it's close to CA2. This was shown also in the Moser lab for place cells in rats. So we found the same thing in our case, but actually for the representation of the other, it was the other way around. So there was interesting difference in functional anatomy there. Yes?
I'm wondering that you don't see dramatic difference between the informative and non-informative.
I agree with you. I thought that we should see, in fact, we had this idea that maybe we will see these agency neurons, right, that will be clustered together the representation of the other bat and or demonstrated bat and demonstrated object. You'd think that way, but in fact we found only maybe a couple of neurons that, if you really squint hard, look like that. But the majority look different. So yeah, I agree it's surprising, but it is what it is.
So the demonstrator is going to produce sound, right? Whether
yeah.
your object, your control object, will be purely visual.
Right.
So it might be that the observing bat is just hearing what the other...
It could be, yeah. There are many differences. They're emitting odors, right? But this is part of what makes them live animals, right? So it could be one of the factors that go into this. Yes, I agree. So let me just summarize this part and then summarize the entire talk, because I think we're running late. So these results demonstrated the position of other individuals is explicitly represented in the mammalian brain. Social places operate in allocentric, not egocentric coordinates, and the representation of conspecific is different from the representation of moving objects. I also like to point that there is a parallel paper from a group in Japan, Fujisawa lab, that found similar things in rats. They did a different experiment, different design, but again, the idea was that a rat had to pay attention to the position of the other rat, in their task, and again, they found neurons that represent the other position. The papers are going to be published back to back. So I think it's nice that parallel things are being found across species. And lastly, kind of on a speculation side, I think that the social place cell may be important for social spatial recognition mammals, right? Representing the position of conspecifics will also be very useful to represent the positions of predators, prey, all sorts of important stuff out there. It's very, you'd like that to be represented, and indeed we found that, yes.
Did you record always from the same hemisphere or in both hemispheres?
Always the same hemisphere.
Which was?
The right one.
The right hemisphere.
Yes. Most hippocampal studies are done unilaterally for historical reasons, but yeah. I have not much to say to defend this, but once that's what most people do, we also do the same in the bats. Okay, so to summarize, this is my last slide. Why do we study bats? I promised in the beginning to tell you. So kind of summary, in a way of summary. So, really we try to use the interesting behaviors of the animal to ask questions that are maybe difficult to ask in other animals. So for example, this is something I really had no time to go over. The bats have this interesting sonar system, very discrete temporal information, sensory information that's coming in that we can measure relatively easily by use of microphones, either ground-based, or now we have this this audio logger that we use to record on an animal. But in any case, we can record the discrete sensory inputs, and it's something that's much more difficult to record in a mouse or a rat as it runs around in some maze. When exactly did it see something, touch something, smell something? It's a difficult measurement problem. And here we can use sound and measure it very precisely. And it turns out this precise timing actually affects place cells in an interesting way. I didn't have time to show you, but it's something we published in the past. You can ask questions about 3D, more difficult to do in rodents, but although I showed you that there are beautiful head, no, I didn't show you, but I told you that there are beautiful head direction cells also in mice now. Theta oscillation, which are really, we had several studies on that, and another one in the revision, but so we have a whole different branch looking at this issue, but it just shows you the power of the comparative approach. It really allows you a direct test of computational theories of hippocampal function, turns out that we see a lot of things that are very similar, but also some things that are different. These allow you to constrain the possible space of theories. They have excellent spatial memory. They are highly social mammals. They exhibit mimicry and observational learning, so we are going more also in the social directions in the near future. And we'd like to bridge the gap between behavior in the lab and natural behaviors in the field on a scale of kilometers. These are wild animals, so we can compare directly, or maybe indirectly, but in the same species. And the last 2 points that are really the most important for me that I'd like to make as a general point. One is the issue of the comparative system in neuroscience that I already mentioned a couple of times, but I think it's crucial to compare across species because especially in the hippocampal field, historically, 99% of what we know about the hippocampus physiologically comes from just one species, the rat, and a little bit in the mouse. There are very few studies in other species. So I think it's crucial, this is a sanity check, to look at other species. And it turns out that a lot of things are very similar, but there are some major differences. Really, this allows you to constrain, to understand what is general and what isn't. So I think this is really, I mean, obviously this crowd is it's preaching to the converted, but I'm also saying this to less converted crowds. And I think it's important to study across species, at least to some degree, to understand what is general and what isn't. And the point that's even more dear to my heart, and this actually has to do a lot with what this entire course, I think, ideology, is this comparison of natural behaviors versus reductionism. So, Western science for the last 400 years has a very strong emphasis on the reductionist approach. You isolate all variables and manipulate only one, and then you think you understand the system better, which, is, if you do physics, is the right way to go, the controlled way. But then when it comes to behavior, my worry always is if you oversimplify things, you constrain the animal too much, you control too much, and you might miss the very thing that you are trying to understand, which is behavior. So I think we really need to go in the kind of electrophysiology world to more natural behaviors. So I showed you in our own work, we went to 3D, we found 3D representations, we introduced goals, we suddenly found representations of goals, we introduced another animal, we found representation of another animal, we go to larger scales, we find completely different stuff. So every time we go a little bit more naturalistic, we find surprising stuff about the brain. So I think this is really important. Of course, there's a lot to be said for reductionism, for control. We have to struggle every day of how do you associate behavioral variables, etc. It's not easy. So I think the really good way is to have diversity, both a reductionist, highly controlled approach, but also a more natural approach. This is the best way to advance. So this, I'd like to thank lab members, especially Liora Lass, who's a staff scientist working with me. She's really fantastic and has been involved in a lot of the things that I told you about. And the students, I mentioned most of them as I went along, and alumni and collaborators and funding sources. And thank you for listening.